Hello, I'm Matt,
And I'm Carmen.
and today we'll be talking about ATP synthase, also known as ATPase, both names which we'll be using interchangeably throughout this presentation. Now, ATP synthase is a protein and enzyme that is classified as a translocase. Translocases, the seventh classification of enzymes, are responsible for moving substrates from one side of a membrane or gradient to another. ATP synthase is a complex protein that can be found in a wide variety of organisms, and in a variety of organelles and cells. The most well-known location of this protein is in the inner mitochondrial membranes of eukaryotes and in chloroplast membranes of plants. This protein-enzyme complex can also be found in the plasma membranes of bacteria, in vacuoles of eukaryotes, and in membranes of organisms in the domain archaea. ATP synthase has several structural variants across the broad range of biological organisms. However, the components thereof are generally the same.
Right, so the ATP synthase enzyme has many components to it. The main two regions are F0 and F1. And the F0 is in the inner membrane of the mitochondria, and it consists of the A and the C subunits. So the A subunit, it has two hemichannels, which are hydrophilic, and one hemichannel is open to the intermembrane space, and the other hemichannel is open to the matrix. This C subunit is like a turbine. It has many C subunits in a circle, and it varies, but it's usually between 10 to 14 C subunits. So that's the F0 region. The F1 region has many more subunits. So starting here, the epsilon and the gamma subunits, these will move according to the C subunits and will change the conformation of this hexamer made up of alpha and beta subunits. The beta subunits have the catalytic sites. The alpha subunits will influence these catalytic sites, and we'll talk about this in a bit more detail later. This is held in place by the delta subunit and connects to the A subunit by this B subunit. And this B subunit holds this F0 and the F1 together. There are three subunits that rotate, which are the C subunit, the epsilon, and the gamma subunit. The rest are stationary. So this is just the beta subunit. And this is the alpha helices. It's worth mentioning that all of the ATP synthase is made of alpha helices. There is no beta sheets in this enzyme. So here in blue is the catalytic site in the beta subunit.
Thank you. Now, ATP synthase acts as a complex chemical motor enzyme by coupling the movement of ions across gradient with catalytic synthesis and hydrolysis. As Carmen described earlier, the F0 component of the protein structure consists of a rotating C ring and an A subunit that contains two proton channels. Protons, typically hydrogen or sodium, depending on the species, will flow through the proton channel and into the C ring. The C ring contains ionizable amino acid residues, typically either glutamic or aspartic acid, and the A subunit consists of an arginine residue that resides next to each proton channel, so typically around this region and this region, both going in and out. The incoming hydrogen or sodium ions will hop towards the negative residue of the C ring, which will then free it from the residue. Will free the residue from its interaction with the A subunit, thus effectively shifting the whole C ring forward, and this will move the proton along with the ring. Now, this whole process happens sequentially, so with the arrival of a new proton on the one side is coupled with the leaving of a proton on the other side to the extracellular space. And so that this way, a new ion pair is formed with the next copy of the C subunit on the ring. This rotary single-file ion transport effectively moves protons from one side of the membrane to another. And as the C ring rotates, it also rotates the gamma subunit, like two gears moving in synchronicity. This rotation of the gamma subunit affects the F1 complex, which Carmen will now discuss.
Right, so in the catalytic sites in the beta subunit, there are many amino acids. We've seen that threonine, lysine, and arginine are common in all ATP synthase. And their role is to bind to the substrate. So there are 3 catalytic sites, and in each site there are 3 states. There's open in red, loose in green, and tense in blue. So an open state here, it's high affinity, and inorganic phosphate and ADP will enter, and also ATP can exit. So once it's entered, and there's a change in a rotation in the gamma stalk, it's going to change to a loose. Again, the hexamer doesn't rotate, it's just a conformation state. So once it's in loose, the ADP and the inorganic phosphate will tighten, and the loose has medium affinity, and it's not going to let go of the ADP and inorganic phosphate so easily. Another gamma rotation will cause the conformation state to tense. It's not seen here in this part, but if it was, it would have been like this. So in this part, in the tense state, ADP and inorganic phosphate tighten together and converts to ATP, so you can see here. But it does not let go of the ATP. Another gamma rotation needs to occur. It needs to change to an open state. You can see here that the ATP has exited.
Now, just like any other protein or enzyme, ATP synthase is regulated by the host organism. While there are over 250 compounds that have been found to have inhibitory effects on ATP synthase, we will only be examining a few endogenous inhibitors. The first of the few inhibitors is IF1. Now, IF1 is a non-competitive reversible inhibitor protein that binds to the F1 units to form a complex that inhibits hydrolysis of ATP. The IF1 protein initially binds with the complex by insertion of its C-terminal alpha helix into the interface between the alpha and beta subunits of F1 ATPase in a mostly unfolded form, and requires the hydrolysis of 2 ATP molecules to fold itself into a form that will actually inhibit ATP hydrolysis. And you can see that here. Here's the protein in its unfolded form. It will begin to fold, but then it will insert itself here into the complex, and then with the hydrolysis of ATP molecules, it will complete its folding and block ATP, or rather the substrates, from binding to the complex. Now, IF1 does not actually inhibit ATP synthesis, but it is a very useful tool for inhibiting wasteful ATP hydrolysis that normally occurs with acidic conditions within the mitochondria. Now, a second known endogenous inhibitor of ATP synthase is angiostatin. Angiostatin is a naturally occurring protein that has been found to block the growth of new blood vessels in mammals, but has also been found to act directly on ATP synthase and will affect its processes. So angiostatin binds reversibly and non-competitively to alpha and beta subunits of ATP synthase and blocks substrates from binding to the complex, particularly during hydrolysis. Angiostatin has been shown to block ATP synthase in the epithelial cells of umbilical cords. However, the full process behind this isn't fully known yet. Now, another known endogenous inhibitor is enterostatin. Now, it kind of functions similarly to the other molecules mentioned, but it's a naturally occurring pentapeptide that comes into play during dietary fat digestion. So it also binds to the beta subunits of ATP synthase, but actually blocks ATP synthase, and thus will inhibit ATP synthesis. Now, just to shift gears a little bit, as you can tell, ATP synthase is an obviously complex protein, and as such, it's derived from a variety of DNA sequences. There are several genes across several chromosomes that are responsible for coding for the various subunits of ATP synthase. However, we will focus on one of the most important subunits, the beta subunit. The genetic code responsible for the synthesis of the beta subunits can be found within the gene ATP5B on chromosome 12, at least in humans. This gene is 660 nucleotides long, as pictured here, and leads to the amino acid sequence that is also pictured here. Now, keep in mind that this is the genetic information that codes for only one of the subunits. So try to imagine the total sum of genetic code that's responsible for coding for all of these subunits. And now, Carmen will talk about mutations and structural variants.
Great. So ATP synthase has many variations, and in structure, it really depends in between organelles, cell type, and organisms. So even though there's a lot of variations in structure, the amino acid residues that are responsible for catalytic activity do the same function, and the purpose remain the same. So for mutations in the mitochondrial DNA, there are two genes, ATP8 and ATP6, that are responsible for the assembly of ATP synthase, especially the F0 region. So a mutation in the ATP6 gene results in Reif syndrome, which results in delayed development and weak muscle movement in childhood or in infancy. It can also result in NARP syndrome, which stands for neuropathy, which is peripheral nervous system damage. It can also result in ataxia, involuntary muscle movement, and retinitis pigmentosa, which is damage in the back of the retina, resulting in vision loss over time. So that was ATP6 gene mutation. An ATP8 gene mutation will result in infantile hypertrophic cardiomyopathy, which is a heart muscle disease in infants. And so these mutations are pretty severe, and they're affecting early childhood or infants. And we don't know the full details about these, but through bioinformatics, we can sort of figure out what solutions there are. So for example, ATP6 gene, if there's a mutation here, they found that there is an imbalance of negative charges in the proton channels. So many drugs are being discovered to treat these sort of diseases caused by the mutations.
And now to shift gears again, we'd like to talk for a little bit further about the function of this enzyme. Now, as we discussed earlier, ATP synthesis occurs with the gamma subunit rotating clockwise. And for the most part during our research, we found mostly information about ATP synthesis and how the gamma subunit did rotate clockwise. So while we were doing the research, we predicted that the opposite reaction, ATP hydrolysis, would naturally occur with the gamma subunit rotating counterclockwise. Now, in order to determine whether or not our prediction was correct, we delved into a variety of papers, and we found our answer in the paper titled Direct Observation of the Rotation of F1 ATP Synthase by Hiroyuki Noji et al. So in their experiment, the researchers took a subcomplex of alpha, beta, and gamma subunits from E. coli and fixed it to a glass plate. Then they attached a fluorescently labeled biotinylated actin filament to the gamma subunit, as you can see here, that would be able to be observed for rotational movement. So at that point then, ATP was infused into the flow chamber containing the subcomplex, and the ATP hydrolysis process was observed with an epifluorescence microscope. With this process, they were able to directly observe the rotational movement of both the actin filament and the gamma subunit, and found that they both rotated in a counterclockwise direction. which then of course confirmed our hypothesis and lends credit to the machine-like efficiency of the multi-purpose protein ATP synthase. So unfortunately, this is all the time we have now for this presentation. Thank you for listening. I hope you learned a little bit about ATP synthase. Thank you. 14 minutes.
Yeah.